composition of air that you breathe right now that we are breathing is composed of nitrogen. It's composed of oxygen, and there's some trace elements, right, that are also in the atmosphere that you see here, right? Now, what is the predominant component of the atmospheric air? Is it oxygen?
Nitrogen.
Nitrogen, right? And you notice it's under pressure. So like, how did they get these numbers? They essentially, if you put them under a piston, right, you'll measure, right, 760. We talked about 760 millimeters of mercury, right, or 760 torr. And then they saw the individual components. Now, is we said the biggest component of the air you breathe in is 78% nitrogen. Only 21% is oxygen. Oftentimes you guys will hear, oh, he is on room air. So you kind of start to think, okay, he's 21% oxygen. Now, is this nitrogen in my lungs as I'm sitting here? Like I'm talking to you, right? Do I have nitrogen in my lungs?
Yes.
Yeah, absolutely, right? So I definitely have nitrogen. I have oxygen there. And the pressures you'll see here. What's important about this concept? Like, who cares, right? Okay, so it's nitrogen and oxygen, but why do I care?
Because if we don't have nitrogen, we'll be burned. If we'll get burned.
Yeah, burn.
If we don't have the nitrogen.
Let me rephrase it. How is this value of room air or atmospheric air being 78% nitrogen and only 21% oxygen affect you, for example, during a procedure such as intubation?
Isn't oxygen like unstable, and so too much oxygen can be bad for you?
It can if it dissociates into free radicals, but usually oxygen is a diatomic molecule, right? And it exists as O2 in the atmosphere. So in that sense, it's stable from that perspective. You guys tell me, did you guys have the airway lectures for intubation or not yet?
We had it.
Okay. So did you learn about the concept of pre-oxygenation?
Yes.
Yes.
Okay, excellent. Before you intubate somebody, you guys learned about pre-oxygenating. So if we are pre-oxygenating, we are providing 100% oxygen through our BVM, right? And my then question, if you know that you're pre-oxygenating, you're providing 100% oxygen, and you know room air is only 78% oxygen. What's the purpose of pre-oxygenation? What are you trying to accomplish with that?
You oversaturate them because they're going to go an extended period of time without any oxygen.
Yeah, but
So
what does it do specifically to this composition here in the lungs?
it changes its volume of oxygen. Yeah.
It's going to increase. And what happens to the nitrogen?
Decreases.
Yeah. So the whole concept, and this concept is very important. I see it very seldom understood, right? One of the reasons we pre-oxygenate is we want to do nitrogen washout. So when I pre-oxygenate, I want to remove all this as much as I can to the point where this becomes 100%, right? I'm going to show you slides and I'll explain to you why is this important, right? But one of the reasons we pre-oxygenate patients prior to intubation is that I want to have a nitrogen washout, right? Because we know that room air, atmospheric air is nitrogen 78%. I want to remove all that. I want to denitrogenate their lungs. So remove all the nitrogen so that I have 100% oxygen, right? So this is just all the guesses here. They'll show you the makeup, right? The percentages of it. And the summation of it, right, will essentially yield 760 millimeters of mercury, which we already talked about, right? And the way, they get these pressures, essentially, they use the piston. This is the piston, right? And under high pressure, right, Henry's law, right, will say as you increase the pressure, the more of the gas will go into the solution, right? If you remove the pressure or you decrease the pressure, you drop the pressure, right, the gas will come out of the solution. So in the solution being, right, in our blood, these gases are under pressure, and that's the pressure that they exert, right? Collectively 760, right? And as far as right, the movement of this, in terms of expiration, we're going to talk about after a break. So what I'm going to do is let me pause. All right. So now we're going to talk about the actual transfer of the gas particles, right, as they travel through your blood, right? So the principle here is also, as I said, for the movement of them is from high to low pressure. So here you will see, right, this is the alveoli, and you notice the pressure of oxygen is 100, pressure of carbon dioxide is 40, but as it comes through the veins, right, the oxygen here is 40 because the tissues picked up the oxygen, and the hemoglobin and the bicarbonate system has picked up carbon dioxide. So as they travel to the alveoli, right, you'll notice that O2 is low, but O2 here is high. So the oxygen will want to go from high to low pressure. And then you notice the CO2 is here is high and here it's low. So the CO2 wants to diffuse out, right? And then here, as this exchange occurred, as I pointed here, as it goes down, right, the oxygen gets loaded onto the hemoglobin, and now it's 95%, right, loaded, and CO2 has been, diffused out through the alveoli, and then goes forward to the tissues, and it's picked up oxygen from from the alveoli, and got rid of the CO2. Once it comes to this portion here, you notice in your actual cells, the oxygen is low and carbon dioxide is high. Why it's high is because it's using the oxygen and it's producing the CO2 through respiration, right, cellular respiration. And you'll have the following exchange, right? Oxygen will load and the CO2 will come in. How this all occurs, I'll explain to you. And this is very important to understand. This is just the bigger pictures of the same stuff. So now we get to the main component of this that is important to understand, right? So the nitrogen, oxygen, and carbon dioxide, they're nonpolar. So if these gases that are, you're breathing in, they're nonpolar, that means that they can just easily diffuse into the blood and, travel by themselves in in that fashion. There has to be a carrier molecule or carrier protein for them, right? As opposed to, let's say, ammonia or hydrogen sulfide, they're polar, right? So they have these hydrogen bonds and they create these charges, creating a polar, essentially, connection, right? Or a dipole moment. But these molecules cannot. So we need to have a carrier for oxygen, carbon dioxide, specifically, right? So what does carry that? And what carries it is this molecule called hemoglobin. Hemoglobin is a molecule composed of 4 subunits. So you have the beta subunit. So you have beta 1, right? Beta 1 and beta 2 subunit. And then you have the 2 alpha subunits, which is alpha 1 and alpha 2. They will cross-link together to make this quaternary structure, right? The essentially hemoglobin is known as a tetramer, right? So you have 1 monomer, 2nd monomer, 3rd monomer, 4th monomer. So these are all monomers, but when they come together, they form a quaternary structure. Essentially, we call it hemoglobin, made up of 2 beta chains and 2 alpha chains. In every single monomer, what we see this ring, right? This ring is called protoporphyrin ring when the iron is not in there. The moment we put iron in there as a metalloprotein, right? The metalloprotein iron is now called a heme, and that's why they call it the heme groups. So now this group, once it's loaded with iron, it's called heme. What's important, right? The most important thing here you see, right? Oxygen specifically binds to the iron. It doesn't bind anywhere else on this molecule. And based on this picture that I said, there is 4 monomers. Each one, right? Each one of these has this heme group, right? How many molecules of oxygen can one hemoglobin carry?
4.
4. That is correct, right? So one molecule can carry 4 oxygens, and the binding of them is cooperative. So what that means is that initially, right, as the first one binds, the moment like it sits in here, it allows easier binding for the second oxygen here. And as the second oxygen binds, the third is easier to bind, and so is the fourth. So they cooperate with each other. It's called cooperative binding. So with subsequent oxygen coming in, it becomes easier for more oxygen to bind. Same thing when they dissociate. Initially, it's hard to get the oxygen off, but as one leaves, right, it becomes easier for others to do that. This is just shows you the actual heme group, right, the iron, where the O2 binds, and this is the iron in ferrous form, right? You see it's bound to nitrogen at these ends, and at these ends it will be essentially will bind oxygen. This end will bind to histidine, the proximal histidine, and this will bind to a distal histidine. So why am I showing you all this, right? And the reason why I'm showing you all this is that imagine you have a molecule CO. CO is what?
Carbon monoxide.
Carbon monoxide.
Carbon monoxide, right? So this is your normal structure, right? You see the oxygen is here, right? O2. So it's a diatomic molecule. And now you have something known as carbon monoxide, right? It's a colorless, odorless gas that you can inhale. And where do you think that carbon monoxide wants to bind?
To the hemoglobin.
Not the hemoglobin, but specifically the iron, right? And the reason why I want to point out is that this portion here, this does not bind CO2, this doesn't bind hydrogen, this doesn't bind bicarbonate. This iron, preferentially, it wants to get O2. But if CO comes in, this will bind more stronger. This actually binds 200 times stronger, right, than O2. And O2 and CO, carbon monoxide, right, will bind here at the iron. But CO2, right, hydrogen ions and everything else does not bind at this location. This is why I have this here, right? And why, you will say, how come the oxygen, binding is not as strong as carbon monoxide? And the reason why is basically the shape it takes. So when oxygen binds the O2, it has this like bend to it. When the carbon monoxide binds, it's straight, right? So the angle of the bond makes steric effect. So here, the histidine that comes in from the top to essentially close out that bond, it has steric hindrance from this carbon monoxide, versus with this oxygen, which is bent, it has very good hydrogen bonding. So when this is occurring, right, this is normal oxygenation that we want, but when we have carbon monoxide exposure, the histidine doesn't bind as much. And more importantly, oxygen can no longer bind here because this is 200 times more, has more binding affinity to this iron, right? So what you need to be aware of, right? If you have someone who you suspect of carbon monoxide exposure, and your meter is going off, you want to take them out of the environment and give them high con O2. You may consider taking them to hyperbaric chamber, where they could change that ATM pressure, right? They could drop it down and load you up with much more oxygen, hopefully to get this out and load you up with O2. But the reason why this is occurring is as follows. CO has a stronger connection because it has this basically bond that does not have any, it has a stronger connection and it has a steric hindrance for this histidine. So histidine does not want to bind here, how it does with oxygen. So this is why you have carbon monoxide poisoning if this becomes exposed too long. So this is the hemoglobin with oxygen, right? And we're gonna get into hemoglobin curves in a little bit, but just to show you, right, the subunits. So this is here your iron, right, bound with oxygen. This is your beta 2 subunit. This is not beta 2 receptor, just to clarify. This is beta 2 globin chain of the hemoglobin. It comes together with alpha 2 chain. Beta 1 comes with alpha 1 chain. This is not receptors. This is just the components of the hemoglobin, right? And what happens here, right? In the lungs, it gets saturated with oxygen because it's under the high pressure. As it goes to the tissues, it will unload. And there is allosteric sites, not here, not here, not here, not here, that will pick up CO2, right? On the hemoglobin itself, and then take it back to the lungs to exchange. But this site's very important, right? This iron is only for binding O2, or in case of poisoning, right, it will bind the carbon monoxide. But do not confuse this binding with CO2 or bicarbonate system or hydrogen system. Right, we are gonna get to this hemoglobin dissociation curve, and this becomes extremely important for you guys when we talk about this pre-oxygenation concepts that I talked about, right? So you notice, right, you have hemoglobin, which is in your red blood cells, erythrocytes, and has the sigmoidal shape. And myoglobin, right, it doesn't have the sigmoidal shape, right? Myoglobin is found in your muscle. It's basically, think of it, if I take the hemoglobin, the 4 subunits, right? I basically take one subunit, that's your myoglobin. Myoglobin is singular, and it binds oxygen, but it doesn't readily give it off as easily, right? This also what gives the red meat its color. So if you see the, beef, right, that's not because it's bloody, it's myoglobin, it's in the muscle, right? So, and it binds oxygen tightly, doesn't give this off. But hemoglobin, you notice, right, it has a different curve at a different portion, right? A good way to remember this, at about pressure of 40, right? This is at the tissue level. Remember, we said oxygen was about 40 at the tissue level, and at the lung level, this was about 100%, or you could say 95%, right? So correspondence is here, right? Let me just move it over. So this is what's occurring at the tissue level, and this is what's occurring at the lung level, right? And why is this important? This is important is that at the lung, you notice this curve it doesn't drop right away. It has this like long shape, and the moment I get to the tissues, it has this steep drop. So it means and here it's the saturation. If I put a pulse ox on your finger, right, like a probe on your finger, and I measured your saturation. So here, if I look, your saturation will be 100%. If I looked here, it will be 50%. So it means oxygen is being unloaded. So you'll say, well, who cares? Why is this important? And once I get further on, you will see how this is important, right? So all of this, right? This oxygen carrying is done by this hemoglobin, right? With alpha and beta subunits where it binds. This is the overall picture of it, right? You see the iron, you see the histidine, right? And you see where the oxygen binds. When this binding of oxygen actually occurs, right? What happens is this oxygen kind of pulls of the negative charge kind of pulls this group, and that what gives this red blood cells, it's kind of like the curvature appearance, right? When it's loaded, so it kind of pulls on these helix F structures, right? And here you see what happens is as oxygen is loaded up on, it goes from the tense state, right, to the relaxed state. In the tense state, this hemoglobin holds onto oxygen very strongly. It doesn't want to give it off. But and the relaxed state is when it wants to get rid of this oxygen. It wants to deliver its oxygen. So we're going to talk about this, right, as it gets to the the cells. I want to show you because I think visually it looks cool, but more importantly, I think visually you kind of get the ideas before maybe you're not. So what this is, there's a site called Protein Data Bank, and this is human hemoglobin, right? They basically put it in oxygen for 20 seconds and how it gets loaded. I'm going to show you, let me show you how this looks, right? So I'm going to stop share. So you guys see the hemoglobin? Yes?
Yes.
Yeah. So here, right, this is the actual molecule, right, and the spatial arrangements of all the atoms, right. So red is oxygen, red atoms. The cyan color is carbon. Blue is nitrogen, right? So, and now let me show you. So this is the entire molecule. Now I wanted to display the heme groups, right? The heme groups that I talked about, right? So these are the 4 heme groups, and in the center, the pink, right? The pink being the iron, right? So the iron is in the center. We see there's 4 of them, and oxygen, right? Here that I pointing on, right? This is this oxygen that will bind here. So let me remove it and show you. So this oxygen, as it goes through the lungs, it will bind to this iron here, and then this histidine molecules will complete it, right? So they will bind on both sides of it to complete the structure, and this, right, this entire structure that is circling right now, this will be your hemoglobin that's, you know, being transported. So I'll show you how it looks overall, right? So your red blood cells are predominantly, you know, bags of hemoglobin. Roughly for a healthy adult person, they have about 15 grams per deciliter per 100 mLs of their blood, right? They have this hemoglobin that's loaded with oxygen that's traveling in your body, going to the tissues. And when it gets to the tissues, what is going to happen is essentially this heme component, right here, right? This oxygen right here will be delivered to the tissues, and this pink part, right, is not involved in any carrying of CO2 or hydrogen ions or anything like that. This is strictly for oxygen. And unfortunately, when carbon monoxide binds, that's the site of the connectivity. All right. So let me go back to my slides. So now that you see how it looks, right? Where does carbon dioxide transport? So carbon dioxide made from your tissues is actually binding to the hemoglobin, specifically the amine component, right? On the globin chain itself, is going to bind there, and then together they call it carboaminohemoglobin. It binds on the hemoglobin, not at that iron site, and then this hemoglobin molecule will transport carbon dioxide right to the lungs. Here you basically see how it does. Unlike, right, remember we talked about the carbon monoxide. Do not confuse it with carbon dioxide, which is CO2. So it binds to the carboamino terminal, right? And this equation is very important for you to learn. This is basically this whole buildup was to this. So what we have is we have CO2, right? Carbon dioxide made by cellular respirations in your tissues, right? And your plasma, right, comes with water to form carbonic acid, which can further dissociate into carbonate ion or bicarbonate and hydrogen ion, right? So this formula, if you see all those arrows, it can go either way. It can go this way, right? Or you could go this way, depending on what the body needs. So just to give you an example, let's say you had a guy, right, who was in respiratory acidosis. Let's say he is under influence of heroin, right? So if he is not breathing, right, let's say he's not breathing adequately. Let's say he's breathing 6 times a minute. Do you expect his CO to go up or down? So I'm not moving much air.
Up.
It's going to go up.
Right?
Yeah, it's going to go up. We know normal carbon dioxide 35, 45. So it's going to go up. Let's say for this guy, it becomes 68. Let's just say. This reaction will start moving towards this side. So it's going to make more. This carbon dioxide that's retained is going to form with water, make more carbonic acid, which is going to dissociate into hydrogen ion and bicarbonate. This will essentially, this hydrogen ion will drop his pH. So this guy becomes acidotic. right? On the other hand, this brand new EMT comes in, right? He's like, yes, I heard about this, concept of positive pressure ventilation, right? This guy is not breathing. Let me ventilate this guy at a rate of 36. So he starts to squeeze the bag 36 times a minute because his adrenaline's through the roof and he wants to ventilate this guy who's not breathing. What do you expect to happen to this carbon dioxide? It's going to go up or down?
Down.
Down. So carbon dioxide goes down. So then the reaction has to replenish the carbon dioxide. So I have to go this way. So I'm combining bicarbonate, hydrogen ion in my blood, making carbonic acid, and I'm basically coming in to replenish all this carbon dioxide that's being eliminated by the lungs. And if this prolongs for a while, my pH is gonna go up, and I'm gonna be in alkalosis. Right. So far makes sense. This portion here, the bicarbonate system, the CO2 removal is usually occurring at the lungs. But there's also another component, right? We can actually, the kidneys can remove the bicarbonate component, or we can, adjust this somewhat ourselves. So the carbon dioxide component is like minutes to seconds. The bicarbonate component from the kidneys is more like hours to days. So this is longer, but this is faster, right? So any respiratory acidosis or respiratory alkalosis is fast with the respiratory component. The bicarbonate system here is more like metabolic acidosis or metabolic alkalosis, and it's predominantly by the kidneys, right? And now let's see, like, how is majority of it is transported. So you see, right? You saw this equation here, right? CO2 with water, carbonic acid, hydrogen, bicarbonate. So majority of your bicarbonate, right? Majority of it, right, is found in your arterial and venous blood, right? And it's being carried, right? And it's charged, so it's polar. That carboaminohemoglobin makes the small percentage here. Some of it is bound to the hemoglobin, as I showed you, right? And you do have some of it, right, floating freely, right, in here. So the bulk of bicarbonate ion is carried in your arterial and venous blood, right? This is if you are going to be working a cardiac arrest, you may see, right, if they get ROSC and paramedics decide to give bicarbonate. So sodium bicarbonate is essentially this here. So if I give this drug, right, I increase my bicarbonate concentration, I expect my reaction to go this way. So what you're going to see is if you have your end-tidal CO2 connected to the endotracheal tube, his end-tidal CO2 will spike up because he's making more carbon dioxide. And that makes sense because if we are giving this drug, we suspect someone is in metabolic acidosis because of the downtime in cardiac arrest, right? Their tissues is not using oxygen, so they begin to use lactic acid, and they're in acidosis. And if they're in acidosis, we give sodium bicarb to essentially, drive that hydrogen ion. I want to combine it with my drug so that I get rid of it. And the way I get rid of it is I make it into carbon dioxide. And if you basically put the sensor on the tracheal tube, you will see, right, the end-tidal CO2 will spike up. So maybe they were, had an end-tidal CO2 of 32, and the moment you push this drug, this will spike up to like 64 because they are trying to get rid of it. Right. This is just so you understand that. So hemoglobin, right? We said the carboaminohemoglobin carries about 23%. The rest of, right, bicarb is carried by, right, in the plasma, right? And this is your red blood cells, right? And this is just a close-up view of all the gases and everything that's occurring. So let's see. So alveoli, right? Oxygen partial pressure is 100, right? Your red blood cells that comes in, it picks up this oxygen, it loads onto the hemoglobin itself, right? Now hemoglobin is rich with oxygen, right? It goes to the tissues. In the tissues, they have low partial pressure of oxygen because tissues utilize oxygen for their metabolic needs, whatever activity they're doing. Let's say I'm the muscle, I'm using oxygen, right, for activity. It then goes from high to low pressure from 100, right, to 40. So the oxygen diffuses to the cells, and also, right, it picks up the carbon dioxide that's in the cells. Carbon dioxide is a byproduct, right, of respiration, metabolism, and it picks up in the blood in the form, right. Some of them is bind to the hemoglobin, right, not at the iron portion of it, and some of it is diffused into the plasma. And then when this red blood cell comes into, right, back to the alveoli space, to the lungs, the CO2 gets removed, and oxygen again gets loaded. So it's basically this process that's occurring in our body, right? Now, the same formula, right, that I showed you, right? CO2 plus H2O, carbonic acid, bicarbonate, hydrogen, works on your respiratory center, right? So how does the body knows to increase the respiration? It monitors the CSF, right? It monitors the CSF fluid using this formula, right? All of a sudden, right, if let's say I am not breathing well, right, I'm not limiting my carbon dioxide like the guy who took heroin, my carbon dioxide level is retaining, right? This reaction is going forward. I make more acid, right? And this proton, it acts on this center area, right? Inspiratory area. And it tells me you got to breathe more, but this drug is not allowing me to do that, right? Because the drug is depressing this area. So it doesn't allow me to do it. So essentially the way your respiratory rate is adjusted is through this area, right? Chemosensitive area, specifically in the medulla, and it adjusted based on the CSF fluid. It measures the level of the proton, essentially pH, right? Potential hydrogen. And if this is going up, right, it means you're more acidotic. I got to increase my rate of breathing. If this is going down, right, it tells me I'm becoming more alkaline. So this is the major control of the respiratory area, right, in your brain.